Look here. This is the key to our magical experiment, the genetic engineering vector plasmid from tobacco mosaic virus. This plasmid contains the replication origin of TMV and can proliferate in high copies in tobacco plants. And this CAD gene is our reporter gene. Now, we will use endonucleases to change the plasmid. Next, using an in vitro transcription system, let TMV-CAD DNA transcribe the corresponding mRNA. Look, smear the mixture containing transcription products on tobacco leaves. Then it's time to wait patiently. On about the 14th day, we can harvest tobacco infected by recombinant TMV. The harvested tobacco is dried and stored at room temperature. The experiment continues. Now, according to the conventional way of decocting traditional Chinese medicine, decoct tobacco. Feed the cooled decoction to rats once a day for 7 consecutive days. After a week, the rats are anesthetized to death. Let's take the mucosa of the jejunum segment and detect the activity of CAD. Look here. In the jejunum mucosa of rats with the CAD gene transferred in, there is obvious CAD activity. C14 is dissociated and has a high migration rate. Look at the control group again. There is basically no CAD activity. This result confirms that exogenous genes can indeed be introduced into body cells through the natural physiological process of the digestive tract. This major discovery will open up a new way for the theory and practice of traditional Chinese medicine and can also scientifically explain the various characteristics of traditional Chinese medicine treatment. It has long been found that the treatment methods of traditional Chinese and Western medicine are different, and the effects also have their own characteristics. Contemporary Western medicine uses a single purified chemical molecule, which is taken orally or injected into the human body. The blood drug concentration rises rapidly, but the treatment effect is immediate and easy to excrete. It is transient or corrective. As for traditional Chinese medicine treatment, it is slow-acting, continuous and regulatory. The characteristics of traditional Chinese medicine treatment can be explained from our analysis. It takes time from gene introduction to product generation, so the effect is slow. The introduced gene has a half-life. If the product persists, the effect will continue. Moreover, after the gene is introduced into the cell, the product can directly contact the cell components and produce a regulatory effect.